everyone, welcome to the IIT PAL series of learning. So, today we are going to learn about regulation of gene expression. So, what is gene expression? If we are going to learn about regulation of gene expression, we should know about gene expression. What is the meaning of this word, gene expression? As we know, gene is the functional part of DNA which is going to transcribe and translate. So, how the gene is being transcribed and translated is called gene expression. So, gene expression is a process by which the instructions in our DNA are converted into a functional product such as a protein. So, gene expression is whatever instructions, characters, signals are there in the DNA is going to passed through RNA to synthesize protein. So, first of all, the DNA will transfer its information to form the RNA, and this process is called transcription. After transcription, basically messenger RNA will give the signal for the amino acid to synthesize the protein, and this process is called translation. We have learned in detail about the transcription and translation in earlier session. So, how this protein is being synthesized from DNA, or we can say how the information present in this DNA is converted into a functional product, that is a protein. So, this process is called gene expression. So, we will learn separately the regulation of gene expression in eukaryotes as well as in prokaryotes. But the start regulation of gene expression in bacteria, that is prokaryotes. As we know, a major difference between prokaryotic cell and eukaryotic cell is the presence of nuclear membrane. The prokaryotes do not have a nuclear membrane, so the genetic material of prokaryotes are scattered in the cytoplasm, while the eukaryotes have nuclear membrane which encloses all the genetic material and form nucleus. And this compartmentalization of eukaryotes make eukaryote different from the prokaryotic cell. So, there is different way of control of gene expression in prokaryotes and in eukaryotes. So, here first we will learn about the regulation of gene expression in bacteria, that is prokaryotes. Before knowing about the gene expression and how it is being regulated in bacteria, let us know about some terms. The first is regulator gene. Regulator gene, it is a gene that regulates the expression of one or more structural genes. A regulator gene is that gene which control the expression. Expression means transcription and translation, protein synthesis. So, regulator gene, a gene which controls the expression of structural genes. Structural genes is the gene that forms template for messenger RNA and is thereby responsible for amino acid sequences of a specific polypeptide. So, structural genes are the functional genes which are going to transcribe and translate to synthesize a particular polypeptide chain or a protein. Constitutive genes, these genes are required for the maintenance of basic cellular function and are expressed in all cells of an organism under normal condition. So, genes which are functioning in most of the cells to maintain the basic cellular functions is called constitutive genes. It is also called as housekeeping genes. Moving to the next terminology, operator. Operator is a segment of DNA to which a transcription factor binds to regulate gene expression by repressing it. So, operator is that fragment of DNA where some transcription factor or repressor protein binds to regulate the expression of gene. Repressor. A repressor is a DNA or RNA binding protein that inhibits the expression of one or more genes by binding to the operator or associated silencer. So, a repressor is that protein which binds over the DNA or RNA to inhibit the expression of DNA to synthesize the protein. Now, let us know about operon. What is operon? Operon is one of the system which is found in prokaryotic cell, and it is defined as a functioning unit of genomic DNA containing a cluster of genes under the control of a single promoter. So, Operon model, Operon system is not a single gene, it is a combination of various genes under the control of a single promoter which is being regulated by different repressor genes, and they all together is called Operon system. Or we can say a group of bacterial structural genes. Structural genes means functional genes which are going to synthesize a protein. So, a group of bacterial structural genes that are transcribed together along with their promoter and additional sequences that control transcription is called an operon. So, a group of bacterial structural genes which is going to transcribe and translate to synthesize a protein with the help of some additional sequences is called operon. So, you must remember that operon is not a single gene, it is a combination of various types of structural genes and some regulatory sequences. In case of bacteria, or we can say prokaryotes, there are two types of operon system. The first one is inducible operon system and second one is repressible operon system. Inducible operon system. Inducible operon system is an operon in which the presence of a key metabolite substance induces transcription of the structural genes. Example is Lac Operon. So, inducible means what? Say suppose I am having this marker. So, to use this marker we need a board, so that we can use this marker to write over the board. So, this marker will induce me to bring the board, so that this marker can be used. So, this marker is inducing me to bring the board. This is called inducible operon. In the same way, some of the key metabolite will induces the gene to express themselves or undergo transcription translation to synthesize a protein. One of the example of inducible operon system is Lac Operon. Lac Operon means lactose operon. We will learn more detail about the Lac Operon in the coming session. Next one is repressible operon. Repressible operon system means it is an operon which always transcribes a structural gene unless a repressor is present. Repressible operon means the genes are always transcribing and translated. It does not matter whether the repressor is present or not. So, such type of operon system is being controlled by repressing the gene expression. We can understand in this way. Keep suppose I am having this marker, so I do not need this marker from anywhere because I already having this. And one of the example of repressible operon model is Tryptophan operon system. Tryptophan, as you know, it is an amino acid. So, if a cell is having tryptophan in high concentration, so cell do not need to synthesize tryptophan amino acid. So, in that case the gene needs to be repressed, or it should be in switch off condition. So, detail of this also we will learn about in the coming session. Now, the inducible operon system. Inducible operon system, as I told you, it is the operon system in which a metabolite, the presence of metabolite will induces the gene to express themselves to synthesize a protein. In 1961, Jacob and Monod describes the Operon model for the genetic control of lactose metabolism in E. coli. So, this inducible Operon model, which is found in bacteria, example is Lactose Operon system. This was given by Jacob and Monod. The Operon, the Lac Operon, is a combination of operator and 3 protein coding genes associated with it. So, Lac Operon model, which is an inducible Operon system, it has an operator along with 3 structural genes which will transcribe and translate to synthesize different proteins for the lactose metabolism. So, lactose, it is a disaccharide. As we know, you have already learned in the carbohydrate, lactose is a disaccharide having one molecule of glucose and another molecule of galactose, and both the molecule of monosaccharides are combined with the glycosidic bond. So, lactose is a disaccharide, is one of the major carbohydrate found in milk. So, it is also known as milk sugar. To utilize lactose as an energy source, E. coli must first break it into glucose and galactose, a reaction catalyzed by the enzyme beta galactosidase. So, lactose, as we know, it is a carbohydrate, and carbohydrate is the source of energy for all the living cell. So, the E. coli will utilize this lactose by breaking down into glucose and galactose, which are the monosaccharides of the lactose, with the help of an enzyme beta galactosidase. So, whenever a bacteria will get the lactose from the media, the lactose needs to be broken down, and to break down the lactose, the bacteria needs to express beta galactosidase. Without beta galactosidase enzyme, the lactose cannot be broken down. So, how this beta galactosidase is being synthesized by the genes of bacteria so that the lactose can be broken down. So, the enzyme beta galactosidase, permease and transacetylase are encoded by adjacent structural genes in the Lac Operon of E. coli. If we learned about Lac Operon, we should know about the organization of lactose gene, how the genes associated for the lactose metabolism are organized in the DNA in bacteria. So, you can see in the diagram, this is the organization of Lac genes. Here we have 3 structural genes, Lac Z, Lac Y and Lac X. Lac Z, by the process of transcription and translation, it will synthesize beta galactosidase. Lac Y, by the process of transcription and translation, it will synthesize permease. Permease is an enzyme. Lac X, by the process of transcription and translation, will synthesize transacetylase. So, these 3 enzymes, beta galactosidase, permease and transacetylase, will involve in lactose metabolism, or we can say breakdown of lactose. Now, to the upstream of this structural gene there is operator, upstream to this operator there is promoter. This is the promoter for these structural genes. As we know, the prokaryotes are polycistronic. Polycistronic means the genes for any particular metabolic reactions are arranged together. There is no any gaps or there is no any non-functional sequences between the genes. They are present in a continuous manner. So, for these structural genes Lac Z, Lac Y, Lac X, there is a common promoter, and as we know, promoter is a segment of DNA where the RNA polymerase binds to initiate the transcription. To the upstream of this promoter, there is a regulatory gene Lac I, and it is the promoter for this regulatory gene, and this is the terminator sequence for this regulatory gene. So, this is how the genes involved in lactose metabolism are arranged inside the bacterial DNA. So, beta galactosidase encoded by Lac Z gene, beta galactosidase will break down the lactose in glucose and galactose. Permease is encoded by gene Lac Y. Permease will help the bacterial cell to accept the lactose molecule, and trans-acetylase help in the acetylation of the molecules, which is encoded by Lac X gene. In the Lac Operon, the Lac Z, Lac Y and Lac X genes have a common promoter. This you have seen in the diagram. Lac Z, Lac Y, Lac X have a common promoter. Upstream of the promoter is a regulator gene Lac I. You can see it is the regulator gene Lac I upstream to this promoter. The Lac I gene is transcribed into a short messenger RNA that is translated into a repressor protein. So, this regulatory gene Lac I will undergo transcription and translation to synthesize repressor protein. This regulatory gene will undergo transcription and translation and it will synthesize a repressor protein, and this repressor protein involves in the regulation of lactose metabolism genes. So, let us see what will happen in the absence of lactose, whether the gene will be expressed or not, whether the gene will synthesize the respective enzymes or not. So, in the absence of lactose, what should be there? The gene should express or it should not express if lactose is absent. So, it is a common sense if lactose is not there in the cell, there is no need to synthesize the enzymes, because we do not need enzymes to break down the lactose, because lactose is not there in the media in the cell. So, what happens during this condition to the genes? The repressor binds to the Lac operator site Lac O. The repressor which is being synthesized by the Lac I regulatory gene will bind with the operator site. When the repressor is bound to the operator, the binding of RNA polymerase is blocked. If the repressor is binding over the operator, as you can see, operator is just downstream to the promoter. So, if repressor is binding over the operator, the RNA polymerase cannot bind over the promoter, and without the binding of RNA polymerase over the promoter, the transcription cannot occur. So, in the case of lactose, if lactose is not there in the media, the gene will not expressed. So, when the repressor is bound to the operator, the binding of RNA polymerase is blocked and transcription is prevented and genes are switched off. So, you can know about this with the help of this diagram. In the absence of lactose, there should not be gene expression, there should not be synthesis of enzyme which involves lactose metabolism. So, how it happens? Lac I, that is a regulatory gene, will undergo transcription and translation. It will synthesize Lac repressor protein. This Lac repressor protein will binds at operator site and blocks the operator. And as we know, for the transcription of these genes Lac Z, Lac Y, Lac X, the RNA polymerase needs to bind at the promoter. So, because of the presence of repressor protein over the promoter, the RNA polymerase cannot bind over the promoter. So, if RNA polymerase cannot bind over the promoter, these genes cannot transcribe and no translation will be there and no protein synthesis will be there. So, structural genes will not express in the absence of lactose. I hope it must be clear to you. Now, what if lactose is present in the cell? If the bacterial cell have the lactose, it means they need to express the genes to synthesize the protein, to synthesize the enzymes, and those enzymes will break down the lactose to get the energy. So, in the presence of lactose, there must be expression of genes and genes should be in switched on condition. But how it will be possible? So, some of lactose is converted into allolactose. If lactose is there in the cell, some of the lactose will be converted into allolactose. These allolactose will binds to the repressor which is synthesized by regulatory gene. The binding of allolactose with the repressor will cause the repressor to be released from the DNA. As you have seen in the diagram, this repressor is attached over the operator, but when the allolactase will combine with this repressor protein, there is conformation change in the repressor protein and the repressor protein will release from here. The repressor is inactivated because of the allolactose. Binding of RNA polymerase is no longer blocked. Transcription of Lac Z, Lac Y and Lac X takes place and the Lac enzymes are produced. Genes are switched on condition. You can understand this with the help of this diagram. In the presence of lactose, there must be expression of genes which is going to break down the lactose. So, this is how the genes works in the presence of lactose. These are the repressor proteins synthesized by regulatory gene. When regulatory gene will undergo transcription and translation, it will synthesize repressor protein. Because there is lactose, some of the lactose will form allolactose and these allolactose will combine with the repressor protein and it will inactivate it, and because of the inactivation of the repressor protein, it cannot bind over the operator and whatever repressor protein is attached over there, it will be released because of the conformation change by the allolactose. Now, this operator is free from the repressor protein. RNA polymerase can bind over the promoter, it will move towards the structural genes, it will transcribe these structural genes to synthesize the messenger RNA and then messenger RNA will synthesize a protein, respective protein beta galactosidase by Lac Z gene, permease by Lac Y gene and transacetylase by Lac X gene. So, the genes will be in switch on condition in the presence of lactose. Here is another model of regulating the Lac Operon which involves the role of cyclic AMP. Cyclic AMP means cyclic adenosine monophosphate. CAP, that is Catabolite activator protein which is inactive in nature, will combines with cyclic AMP. The CAP CMP complex get activated and it will binds at cap site of Lac Operon. This complex, that is CAP CMP complex, recruit RNA polymerase to bind over the promoter to transcribe the Lac genes, and transcription is followed by translation, and respective proteins, respective enzymes will be synthesized in the presence of lactose. So, you can see in the diagram CAP, that is catabolite activator protein. It is normally inactive in condition. 4 molecule of cyclic AMP will combine to form CAP-CAMP complex. This CAP-CAMP complex will bind over the cap site of the Lac Operon. This complex CAP-CAMP complex will recruit RNA polymerase. This one is RNA polymerase enzyme, which help in transcription. With the help of this CAP-CAMP complex, the RNA polymerase will bind over the promoter, it will move towards the structural gene, it will transcribe the genes to form the messenger RNA, and protein will be synthesized. And this is how the CAP-CAMP will perform its role for lactose metabolism. Moving to the next, let us talk about repressible operon system. This operon is normally turned on and must be repressed. Repressible operon system means the genes are always expressing itself, and if genes are expressing itself, it must be repressed. Say suppose we have a protein in our cells. So, for the synthesis of that protein, the genes should not be expressed because the cells already having those proteins. So, in those conditions, if certain proteins or certain amino acid is there in the cells, the genes should not express themselves. Example of this repressible operon is Trp operon system. Trp means tryptophan. Tryptophan is one of the amino acid. The Trp operon contains 5 structural genes: Trpe, Trpd, Trpc, Trpb and Trpa. These 5 genes undergo transcription and translation to synthesize different enzymes which will involve in the synthesis of tryptophan. Upstream of these structural genes is the Trp promoter. So, you can see the organization of Trp operon here in the diagram. These are the structural genes Trpa, Trpb, Trpc, Trpd and Trpe. To the upstream these structural genes, here is the promoter, and to the downstream this promoter, here is the operator. These are the leader region and these are attenuator region. We will learn the detail of this attenuator later in the session. So, this is how the genes are being organized for Trp biosynthesis. These Trpa, Trpb, Trpc, Trpd and Trpe will synthesize different types of enzymes. And what are those enzymes? Trpe will synthesize intranilate synthetase 1. Trpd will synthesize intranilate synthetase 2. It is also known as PRA synthetase. These are the enzymes. Trp will synthesize PRA isomerase INGP synthetase. Trpb will synthesize tryptophan synthetase B, that is beta. Trpa will synthesize tryptophan synthetase alpha. So, these are the enzymes synthesized by these genes, and these enzymes will help in the biosynthesis of tryptophan. Moving to the next, what happens if tryptophan is absent in the cell? If tryptophan is absent in the cell, what should be there? The gene should express or not? Yes, you are right. If tryptophan is not there, if certain amino acid is not there in the cell need to synthesize the respective genes which will involve in the biosynthesis of tryptophan. That means the gene should be in switched on condition in the absence of tryptophan. So, in the absence of tryptophan, when tryptophan levels are low, RNA polymerase binds to the promoter, transcribe the 5 structural genes into a single messenger RNA. Messenger RNA translated into enzymes that convert corsimate into tryptophan, and genes are switched on condition. So, in the absence of tryptophan, genes are in switched on condition. How it is being done or how it is being performed by the cells inside the bacterial cell, you can see in the diagram this what actually happens inside the cell in the absence of tryptophan. This TRPR repressor gene, it is the repressor gene, is present some of the distance from these organization of genes. This TRPR repressor gene will undergo transcription and translation to synthesize aporepressor protein. This aporepressor protein is naturally inactive, and because of this inactive aporepressor protein cannot bind over the operator. So, in this condition, in the absence of tryptophan, the operator is free. Now, the RNA polymerase will bind over the promoter, it will move towards the structural genes, these genes will undergo transcription and translation, and the protein will be synthesized. So, in absence of tryptophan, the genes are in switched on condition, and what will happen in the presence of tryptophan? If the cell is already having the enough quantity of tryptophan amino acid, whether the gene should express or not, whether the cells need much more tryptophan if they already having enough quantity of tryptophan. Yes, you are right, there should not be expression of genes because the cell is already having enough quantity of tryptophan. So, what happens at the molecular level? Some distance from the Trp operon is a regulator gene TRPR. TRPR encodes a repressor that alone cannot bind DNA. Binding with tryptophan causes a conformational change in the repressor that makes it capable of binding to DNA at the operator site, which overlaps the promoter. So, you can see here in the diagram, this TRPR repressor protein will be synthesized by TRPR gene. This TRPR gene will undergo transcription and translation, and it will synthesize inactive aporepressor protein. But in the presence of tryptophan, this tryptophan molecule will interact with this aporepressor, inactive aporepressor protein, and do some conformational changes in the protein and will activate it. Now, because of the tryptophan, there is active TRP repressor protein. This active TRP repressor protein will bind over the operator, and it will block the binding of RNA polymerase over the promoter. And if RNA polymerase cannot bind over the promoter, there will be no any transcription and translation of these structural genes, and no enzymes will be synthesized for the synthesis of tryptophan. So, this is how the genes works during the requirement inside the cell. Trp operon also regulated by attenuation. Attenuation is one of the particular mechanism found in prokaryotes, in which the complementary sequences of some DNA will get the information, will get the signal for the pausing or anti-termination or termination of transcription. Let us see how it happens. So, here is the organization of attenuated sequences. Here you can see there are 4 regions, 1, 2, 3, 4. These 4 regions are there in the Lac Trp Operon system. You can see over here, these are the attenuated sequence just to the upstream of these structural genes, and these attenuated sequences are arranged in this manner 1, 2, 3, 4, and to the downstream to this 4 sequence the structural genes for Trp biosynthesis are there. So, now when these attenuated sequences will undergo transcription, the sequences of 1 and 2 are complementary, and where they undergo transcription, they will get attached together by the complementary nature of 1 and 2 sequences, and the binding of 1 and 2 sequences will give the signal to pause the transcription. And if 2 and 3 sequences are being transcribed and they also have complementary sequences, and because of the complementary sequences, if they bind together, they will give the signal for anti-termination, means the transcription will go ahead. And if the sequence 3 and 4 is going to transcribed, after transcription they also have complementary sequences, and because of the complementary sequences, if they are binds together, they give the signal for the termination of transcription. So, this is how the attenuation occurs for the Trp Operon system. So, this is all about the regulation of gene expression in prokaryotes. Let us know about the regulation of gene expression in eukaryotes. As we know, eukaryotes have different compartmentalization. They have nucleus, there is different process in nucleus, there is different process in cytoplasm, and because of that there are different levels of control of regulation of gene expression. In case of bacteria, the only one level is there which can be controlled and the protein synthesis can be regulated. But in case of eukaryote, there are so many regulation, there are so many control levels where the regulation of gene expression can be done. And these labels of controls of gene expressions are messenger RNA transcription control label, processing of pre-messenger RNA control label, transport of messenger RNA to cytoplasm control label, translational control label, messenger RNA degradation control label, and protein degradation control label. These are various control labels found in eukaryotes, and this is because of the compartmentalization, because of the presence of nucleus in the eukaryotes. You can see in the diagram the formation of RNA from DNA is called transcription, and here the transcription can be controlled, and if transcription is being controlled, the protein synthesis can be regulated. But in case of eukaryotes, pre-RNA is being synthesized after transcription, it will undergo processing and mature RNA will be synthesized. This steps will also can be controlled, and this is called processing control label. After the formation of mature RNA, this mature RNA is transported to the cytoplasm, and this transport can also be controlled. After the transport of messenger RNA, mature messenger RNA, it will either go to synthesize protein by the process of translation, and this translation will also control, or it will undergo messenger RNA degradation control. Even after the formation of protein, the protein degradation control label is there. So, these are the different labels of control of gene expression in eukaryotes. If we talk about messenger RNA transcriptional control, this is being controlled by regulatory proteins, promoter and enhancer, general transcription factors, activators, and repressor. All these factors control the messenger RNA transcription, and the protein synthesis can be controlled. Role of chromatin in regulating transcription, which this we learn in the transcription process also. As we know, eukaryotes have histone octamers where in which the DNA is being wrapped. So, what happens to these histone octamer during transcription? These histone octamers are being removed up, and this removing is controlled by two different enzymes. The one is histone acetyl transferase enzyme, and second one is ATP dependent nucleosome remodeling complex. These two enzymes will help in the removal of histone octamer from DNA, so that the DNA transcription can be initiated. So, this is how the chromatin can be regulated during the transcription. The next level of control is processing of pre-messenger RNA control, and it includes alternative polyadenylation site and alternative splicing site. For a particular protein, if there are 5 exons and 5 introns, and the same enzymes are being synthesized by the same genes, but there are 4 exons with the 5 introns. In those cases, there is alternative polyadenylation site and there is alternative splicing site. These can be controlled during the processing of pre-messenger RNA into mature messenger RNA. Next control level is translational control level. It includes RNA interference. Nowadays, there are so many research are going on this RNA interference. So many different diseases are being cured by RNA interference for the welfare of the human being, and this RNA interference includes microRNA, that is miRNA, and short interfering RNA, that is siRNA. These microRNA and short interfering RNA helps in controlling the translation with the help of dicer and RISC complexes. It helps the degradation of messenger RNA. Next to this, messenger RNA degradation control level is there, which includes deadenylation dependent decay pathway and deadenylation independent decay pathway. In this degradation control level, the messenger RNA will be degraded either with the help of deadenylation or without the help of deadenylation. Deadenylation means removal of adenine nucleotide from 3' end. First, in case of deadenylation dependent, all the adenine nucleotide from 3' ends are being removed up. After that, the 5' end capping is being removed and the remaining messenger RNA will be degraded. In case of deadenylation independent decay pathway, there is no any deadenylation, there is no any removal of adenine nucleotide from 3' end, but the 5' end capping will be removed, and because of that the RNase enzyme will digest the messenger RNA. So, this is how the messenger RNA degradation is being controlled. Next is protein degradation control. If the protein has been synthesized and we do not need that protein, so those protein will be degraded, and the degradation of protein is under the control of ubiquitin protein. This ubiquitin protein will bind with the protein which is going to degrade. The combined ubiquitin with the protein is called ubiquitinated protein, are transported to proteasome. Proteasome contains protease enzyme. Protease enzyme means the enzyme which is digesting the protein. There in the proteasome, the protease enzyme will digest the tagged protein, the protein which is going to degrade, which we do not need. It has been synthesized by the cells, by the genes, but we do not need those proteins. So that protein will be digested with the help of ubiquitin and proteasome complex. Now, moving to the last part of this lecture, let us think over this question, find the answer of this question, and the answer of this question will be given in my next session. The first question is, what do you mean by gene expression? You learn in this session the gene expression, so you can be able to answer the question, what do you mean by gene expression? What is Operon system? You have learned in this session about the Operon system, which controls the regulation of genes expression in bacteria. So, you have to know about the operon system. Give an example of inducible and repressible operon system. You learn about two different types of operon system in prokaryotes. One is inducible, in which the genes are expressed only when it is being induced by the presence of certain metabolite, and repressible operon system, in which the genes are repressed due to presence of certain metabolite. So, you have to give an example of these operon system. The last question is differentiate between inducible and repressible operon system. If you know about inducible and repressible operon system, you should also know about the differences between these inducible and repressible operon system. So, with the answer of this question, we will meet in the next session. Till then, goodbye, thank you and have a nice day.